Welcome back to our lectures on fluorescent spectroscopy. This is part 2 of 2 on fluorescent probes. In the previous lecture, we discussed all of the classical fluorescent probes, mostly the ones that were all organic. Today, we're going to describe a few of the more unusual types of fluorescent probes or sensing probes, and we're going to describe some that contain metal ions in them. So first we'll talk about is microviscosity probes. What is microviscosity? The normal macroscopic viscosity is the long range motion of a fluid, through a tube or across the ground or any such thing. Microviscosity is really just the immediate environment around a probe. So if you put a probe in a cell membrane, it's going to reveal the microviscosity of the probe, and it's not going to reveal the macroscopic viscosity around the probes. And we call these molecular rotors. And as a plot here, we have the viscosity of the solution versus the quantum yield. And as you can see, the quantum yield of this particular molecular rotor is highly dependent on the viscosity that it finds itself in. How do these probes work? They work similar to a still beam, which we described in the first lecture. The probe, once it's in the excited state, undergoes some type of motion. If there's no motion, it just returns to the ground state. And by motion, we mean rotation about this bond. Or if it's in a fluid solution, it can rotate around that bond. That brings it to a lower energy state that will decay by radiationless relaxation, and hence the quantum yield gets reduced. On the lower panel here, we have examples of a model membrane that is either in the solid phase or the liquid phase. In the solid phase, the viscosity is higher and the lifetime is longer. In the liquid phase, the viscosity is lower and the lifetime is shorter. So let's look at an example of how these can be used in biology. The example I have here is for some dying HeLa cells. And you notice this is actually a Bodippy type molecular rotor rather than the previous type. And you can measure the microviscosity either by the intensities or by the lifetimes. And if I draw your attention to the middle panel here, you see that as the cells are dying, this is a FLIM image, calibrated to the microviscosity over here. The microviscosity is going up as the cell is dying. I don't know why that's the case, but that's what happens with these particular cells. I found this one sort of interesting because, what is the microviscosity of a protein or a binding site on the protein? And these top values really represent when this particular probe, CCVJ, is completely bound to the binding site. And basically, it says that binding sites of proteins have a very high viscosity, in the sense that the probe cannot rotate in the binding site. And note that a dilute protein solution does not have a high macroviscosity, so these probes sense only that local viscosity. We're going to be changing between types of probes rather quickly. So that was our discussion of viscosity probes. And now I'd like to talk of two other types of probes for you. Some are just for like a mean detection. If you're running a gel and you don't know where a protein is, you could react it with some of these probes. They only become fluorescent once they've reacted with an amino group, so that would tell you where your protein is on a gel. These are a little more interesting, another type of fluorogenic probe that depends on enzyme activity. So depending on the presence of a particular enzyme, the probe will be hydrolyzed. And when it's hydrolyzed, it goes from a dark non-fluorescent state to a fluorescent state. And these are widely used in protein assays, or when you're trying to identify the effect of a drug on a cell. You want to know some enzymatic activity, typically done in a microtiter plate. I couldn't find a fluorescent one, but I found this one that changes colors, which is due to some type of chromogenic reaction rather than a fluorogenic reaction, but that's a typical microviscosity probe. Here's a nice example of one that was done with cells. I presume they were living, but if you dissolve chlorine in water, you form some hyperchlorous acid, and that can react with this particular probe and turn it from a dark state to a fluorescent state. So on the bottom here, we have this same set of cells that were not exposed to hyperchlorous acid. They're non-fluorescent. The bright field image, as usual, doesn't reveal very much specificity about the cells, but the probes that were exposed, or the cells that were probed to the hypochlorous acid, are brightly fluorescent, so you can tell whether that exposure has occurred or not. And like I said, we're going to be moving quickly between probes. Be a little more specific about chloride-sensitive probes. Normally they don't show spectral shifts, but first we'll look at what normally happens. Chloride will quench this particular probe. It's a quinoline probe, and they're usually presented as Stern-Volmer plots, which we'll describe in our future lecture on quenching, but it's really the inverse of the intensity. So as these lines go up, it means the intensity is going down. So you can look at the lifetime, you can look at the intensities of the probe, but that's hard in microscopy because you don't know how much probe is in each particular place. But if you do a Flemin image or lifetime image, you can tell where the chloride is most concentrated, because that's where it is quenched the most. And so that says the chloride concentration is higher. You can see that up here, the higher chloride concentration shifts you to the reddish color. Intensity probes are hard to use, especially in microscopy, because you can't control the local concentration. But one way around that is to turn your lifetime probe into a wavelength ratiometric probe. In this particular case, the fluorescent part, the chloride center, was covalently linked to another probe that was not sensitive to chloride. And then when you look at the effects of chloride on that probe, you'll see the quenching effect on the chloride sensitive part, and the other part of the probe is your reference. As you might remember from the very first lecture, I said, absolute intensities are very difficult to measure, so this is an internal reference. And if you make this type of probe, it avoids the need for lifetime measurements if you don't have the ability to do lifetimes or FLIM measurements. They don't have to be attached that directly. This I thought was an interesting application, where what they did is they attached, this is another chloride sensitive probe. It happens to be more sensitive to chloride than the previous one, but they attached it to dextran, and also on the dextran they put tetramethylrhodamine. So that's their standard. The TMR peak is here, and here's the probe in the presence of increasing amount of chloride. It's being quenched, and you take the red over the green ratio, and that tells you the chloride concentrations. So both of these, the previous slide and this one, we would call them tandem probes. You can make tandem probes for almost anything. And as I mentioned, it's bound to dextran. Switching again to another type of probe, and these are, I think they're getting increasingly interesting. These are the aptamer probes. Aptamers, in a way, behave like antibodies, but they're made out of typically DNA strands, and the sequence is chosen so that the strand is partially self-complementary, but it doesn't become completely bound to the complementary parts until a particular molecule is available. In this case, it was cocaine. So this probe will have a fluorophore on it, and this is actually the acceptor here, which the authors labeled D, but it's really a non-fluorescent molecule. In order to do a FRET measurement, you need the spectral overlap. The acceptor does not need to be fluorescent. So this DNA sequence will close up in the presence of cocaine, and it'll be quenched, and you can measure the cocaine concentration. You can make aptamers to many different types of molecules because the sequence determines what the binding site looks like in the middle. This is a two-part aptamer, and I'll just give you some of my reasoning on this, that this one is likely to be more specific than the previous one for a couple reasons. One reason is it uses excimer formation. So that means the two pyrene molecules have to get very close to one another in order for it to sense whatever the target is. And that's the excimer fluorescence, is the longer emission here. Another part of this that I think is likely to make it more specific is that it's a two-part probe. So two different strands have to come together to form the binding site and have the analyte-induced excimer emission. Okay, on to yet another type of probe. These are lanthanides. And the lanthanides are quite interesting. They have extremely long lifetimes. Typical ones are europium and terbium, but there are some other longer wavelength ones. If these were typical fluorescent probes in water solution, they would be completely quenched by the dissolved oxygen. You would never see them. So that's sort of strange right there, and we'll explain it in a moment. And another unusual feature of these molecules is they have extremely weak extinction coefficients, almost unmeasurable. So the only way you can get a significant excited state population is by having the ion chelated to a sensitizer, and the sensitizer has a fluorescent group on it, and these molecules are actually excited by FRET in the near field from the chelator to the ion. So anyway, there are a couple of other longer lanthanides. I haven't seen very many publications on these, but I think they would be useful, especially if you were doing animal studies with mice or rats, because you could see the longer wavelengths through the skin. So why are the lanthanides not quenched by oxygen? This was a mystery to me for a long time, and I finally read closely enough to figure out why. And that's because all of the luminescence and absorption of these probes, the sensitized absorption, is at a lower level than the outer level s electrons. And these two s electrons protect all of the inner layers from the outside world. And for that reason, they're not quenched by oxygen. And they're not really sensitive to the external environment. So everything, that's why it looks like an atomic fluorescence, because there's no contact with a solvent around it. Where are these very long lifetime probes used? And where they're used a lot is what are called so-called time-resolved immunoassays. Now, the term time-resolved immunoassays is really a misnomer because they're not time-resolved in the way we've described for measuring lifetime measurements. They're time-resolved in a way that they just integrate the emission for a period of time and call that total area or total integrated intensity their measurement. The reason that's done is because all of the autofluorescence from biological samples is at much, much shorter lifetimes. And gating a detector on and off on the millisecond time scale is electronically trivial. So no matter how much background fluorescence you have, it's going to be gone by the time you turn on your counting window and see the lanthanide emission at a longer time. And I like this example of using lanthanides to measure DNA hybridization. And the way it was done in this case is the chelator was put on one strand. So here you have your europium, which will be fluorescent in solution. But then you want to measure the hybridization. So what you use is something that intercalates into the DNA. And when it intercalates into the DNA, it brings it close to the fluorescence lanthanide, and hence you can detect a complementary sequence or DNA hybridization. And once again, the acceptor does not have to be fluorescent. It just has to have significant absorption. Okay, on to the next class of fluorophores. These are the green mice, which are made by genetic modification. And the reason they were made is because they were easy to find in New York in the dark subways. Of course, that's a joke, but this is a great picture of the mice that have been genetically modified compared to their sibling that was not genetically modified. So somehow a gene is put into the mice that makes them fluorescent. And I found this example even more remarkable, making plants fluorescent. These are leaves from a citrus tree, and the modified one and the wild type modified is fluorescent, and the wild type is not. And these are simply side sections. And actually, I believe these are commercially available at this time. So you can buy a fluorescent plant, or at least a bioluminescent plant. I think I saw them on Amazon. So where does this come from? What type of gene was used to make the mice fluorescent? And it's actually from a jellyfish, and that had a remarkable property. Somehow the jellyfish is made this protein, it's a beta barrel protein, and it has a fluorophore inside of it. Now we know that it's not too hard to get a single gene into some microorganism, but to make a chromophore that looks like this is going to take dozens of enzymes, and it'd be remarkable if you could get those dozen of enzymes all to function to make the chromophore. But what is magical about the green fluorescent protein is that it is a autocatalytic process. No one predicted that this could happen, that this was a mystery for quite some time. And it turns out that when the protein falls, when that beta barrel forms, it's an auto-oxidation reaction, and the chromophores form spontaneously. So that was just remarkable and resulted in a couple of Nobel Prizes. But the implications of this are many, because you can put a single gene into almost anything, and surprisingly, that autocatalytic reaction was not all that sensitive to the amino acid sequence. So by substituting different amino acids right in that site, they could make different color GFPs. So even though the GFP can be any color, I'll just call them all GFPs. And by the way, this top one is an absorption, so it's not completely properly labeled. To show how remarkable this is, the xenopus embryo was injected at an early stage with the gene for GFP, and one of from the initial division, one of the cells was injected with GFP and one with a red fluorescent protein. And as the xenopus matured, basically one side of the organism was green and the other side was red. And this means in any growing organism, if you can get the GFP gene into one of the earlier cells while the organism is growing, you can follow the lineage of the cells as the organism grows. So you can imagine how useful that is for embryology. Oh, I was just going to mention, this is something that is interesting to biophysicists, because I won't try to explain this in detail, but the GFP fluorophore is rigidly bound into this beta barrel structure, and this was shown by making crystals and using polarized excitation. We'll be talking about anisotropy and polarization in a future lecture, but this just showed that the probe is completely rigidly bound, which is a surprise, because when we talk about protein fluorescence in detail, we'll see that the tryptophanes are almost always have quite a bit of rotational freedom. In addition to following the growth of an organism, you can make sensors out of GFP. And they're typically FRET sensors, and they can be done in a couple of different ways. But typically you have one molecule that binds another molecule. They don't have to be covalently attached like they are here. It could be a protein, something that protein that changes conformation in response to a ligand. But when that happens, if the central moiety is labeled with two different proteins, that conformational change results in an increase in energy transfer and a color change in the observed emission. Another example here is for, I believe it was an alkaline phosphatase, but same thing. When the center part folded up, the color changes. The probes do not have to be made in this way. You can also make them as two separate moieties to see if one protein binds to another protein. So you make one protein with GFP on it, make another protein with the yellow fluorescent protein on it, and if they bind, you'll see a color change. Okay, again to another change in topic, and these are the phycobility proteins, and this was another surprise from nature. I don't know why anyone would think this, but anyway, in the process of photosynthesis, we have phycobilisomes from some certain type of blue-green algae. These are massive collections of proteins, because even each of these individual proteins are quite large, as we'll see on the next slide. And they collect light and they take it to the central reaction center. What the surprise was is that the phycobility proteins, the isolated proteins from the phycobilisomes, were highly fluorescent and highly stable in solution, much more stable than you would expect. So there's like three different proteins there, and they have different numbers of subunits and different numbers of chromophores on them. But remarkably, they're highly stable, and you can attach them to the outside of cell membranes. So these are very bright fluorophores that you can use in flow cytometry, covalently link them to the outs. They'll be, of course, bound by some antibody to a cell surface marker, and you would see it in the flow cytometry. This is also very interesting, the quantum dots or the semiconductor nanoparticles. Who would have thought that if you take a inorganic material and you make the particles small enough, they become fluorescent? But that's exactly what happened. I thought the initial results on this were very pessimistic because the fluorescence was very weak, but over time the synthetic methods were perfected, and it could make these nanoparticles at a homogeneous size, and they could become highly fluorescent over a wide range of wavelengths, depending on their size and atomic composition. For a single type of atomic composition, this is actually completely explainable by quantum mechanics. In order to be used, they had to be modified a little bit, and this took a few years to get right. And I hope yes, good, I do have it on this slide. If you just use the isolated semiconductor nanoparticle, it's very sensitive to the environment. They're unstable, and the last thing you would want is these heavy metals leaking into some organism. So it was decided to coat them with something else, which in this case was zinc sulfide, and that prevented any leakage of the cadmium out. And it made the nanoparticle a lot like the lanthanides, where the fluorescent part was protected from the outside world. So the quantum yield went up when they put the zinc sulfide around it. And then they had to make it useful for biology. So they put a polymer coating around it and then attached the antibodies or the bioaffinity molecules to it. And I thought it might be interesting to just look at what is the size of the quantum dots compared to proteins. And they're not that different. The quantum dots are larger than small proteins, but they're smaller than big proteins like an immunoglobulin. So they're overall they're pretty small in size. They have some unusual spectral properties, which has really made them useful in the medical community. First thing is they have very narrow emissions spectra. maybe not as narrow as the lanthanides, but they're even more narrow than the cyanin dyes. The other remarkable thing is they have high extinction coefficients. They're about 20-fold higher than any extinction you can get from a normal organic dye. And in contrast to the cyanin dyes, which don't absorb any light in the mid-range, they absorb light at all wavelengths. So they're not sensitive to the wavelength, to the excitation wavelength. And this has some, oh, I forgot to tell you, they're almost infinitely photostable. So if you compare the photostability with even an Alexa dye, which are more stable than the fluoresceins, you see the Alexa dyes bleach out right away, but the quantum dots live forever. Unfortunately, these are color-coded in the opposite directions, but I think you can see that the quantum dots are stable, whereas the organic fluorophores will quickly fade in a fluorescence microscope. The reason that the absorption at all wavelengths was interesting is because it had immediate applications for biomedical applications. Because if you wanted to do a multiplex assay and detect more than one type of antigen at a time, you would have to typically, if you were using cyanin dyes, you would have to have a laser or a light source for each one of the absorption maxima of the cyanin dyes. That's not necessary with the quantum dots because they absorb at all wavelengths. So with one laser source, you can incite quantum dots of very different colors, and this is used for following either the detection of a cancer or the response to treatment for cancer, because if you have a cell and you ask the question, what antigens are expressed on the surface, you could see that by the different emission peaks that come from that cell. Many flow cytometers measure the emission wavelength, different emission wavelengths. And in many types of cancers, the diagnosis of the treatment of the cancer is dependent on not detecting one biomarker, but multiple biomarkers. And this can also be done, on a slide. It didn't have to be done as I showed in the previous slide. And this is remarkable. I would have never thought this was possible, but this had to be done for the flow cytometry groups. If you're going to use a flow cytometer, you want to be able to separate all the wavelengths, and notice this is a log scale. The optical engineers who design the optical interference filters are able to design optical filters with 4 band passes, 3 band passes, whatever you like, and thus detect how much of each of those individual nanoparticles is bound to the cell surface. And this slide was just meant to show you again that if you're using a rhodamine and you also wanted to use some other dyes, you would still have to excite the rhodamine at the maximum because of its low absorbance at other wavelengths. But in contrast, the quantum dots absorb at all wavelengths. Well, that's all I have to say for today, and hopefully I will see you at the next lecture.